Well, thank you very much. It's an honour to be here. And I've been involved in drug discovery since I qualified as a medical student, working initially on African trypanosomiasis and then latterly expanding into other kinetoplastid diseases and malaria as well. And we work in the pre-competitive stage, the early stage of drug discovery in Dundee in the drug discovery unit. And I'd like to try and exemplify how we can work with industry to accelerate the discovery of new tropical drugs for the treatment of tropical diseases. So the mission of the drug discovery unit really is to make use of our excellent knowledge in basic science in Dundee and combine it with pharmaceutical know-how, basically. We're definitely not for profit, and our driver is unmet medical need. And our aim is not to compete with industry, but rather fill that early gap in drug discovery, de-risking compound series such that we can engage then with the pharmaceutical companies. And we do this in two areas, the neglected diseases area, both tropical and orphan diseases, those are neglected diseases of the developed world, and also to take on high-risk areas of potential therapy for major diseases. So the Drug Discovery Unit is housed in the James Black Centre, shown on the left here, and in October we will have the official opening of a new building which will double our capacity for medicinal chemistry and drug discovery. So the objectives in the area of the diseases of the developing world is delivery of drug candidates for preclinical development in African sleeping sickness, leishmaniasis, Chagas disease, tuberculosis, and malaria. And on the way, we hope to be able to identify high-quality leads which can be used by academics as chemical tools to validate particular targets and pathways, and also perhaps engage other drug discoverers in taking on a particular target when we don't have the capacity to deal with it. So we've really heard why we are engaged in this area. There are no efficacious vaccines for the parasitic diseases at least, and vector control and public health measures alone are inadequate. And most of the drugs are simply not fit for purpose, and I think Mary Moran, in her analysis in the new landscape of neglected disease drug development, really highlighted that I think of the 17 drugs she analysed, only 4 were really fit for purpose in settings of poverty. And ironically, those mainly came from piggybacking or drug repurposing of veterinary compounds. Why? Because the target product profile for a veterinary anthelminthic, of course, is driven by price as much as anything else. And it has to be easy to administer for farmers to use. So it's interesting that the major successes have been in that area, whereas other repurposed drugs tend to be anti-cancer drugs, and frankly they come with a large number of potential disadvantages which really do not make them suitable for resource-poor settings. So how do we think we've managed to achieve what we've done so far? And really it's a blend of academics on the left with people who really know what they're doing in drug discovery on the right. And Paul Wyatt, David Gray, Kevin Reed are head of the 3 key elements of drug discovery here. And now we have over 70 staff involved in drug discovery, and collectively have apparently helped to deliver more than 50 clinical candidates, not in the neglected disease area, of course, but in other therapeutic areas. So you can read the key elements for successful innovation, and I'll just highlight the fact that you've really got, if you want to do translational research, it has to be underpinned by scientific excellence. If that's not the case, then you're on a hiding to nothing. So high quality basic science has to underpin everything. And then it's really important that you use the pharmaceutical expertise and run things in a business-like or biopharma-like fashion. So I think that's enough on that. Now, we've received considerable funding since we were founded in 2006, following funding from the Wellcome Trust, principally to work on human African trypanosomiasis. And then, as we progressed, we were able to engage with public-private partnerships such as DNDi and Medicines for Malaria Venture, and more latterly with the Bill and Melinda Gates Foundation. So success breeds success, and it's possible then to expand into new areas. But we started very focused, and now we have a number of different portfolios underway. The thing that is quite striking about this university drug discovery setting is that actually we have full capabilities for drug discovery up to candidate selection, in that we can carry out high throughput screening. We have various types of compound sets. We can use target-based or phenotypic approaches. We have industry standard data management systems. And importantly, amongst the basic scientists, we have structural biology. Within the DDU, we have computational chemistry, and of course, extensive medicinal chemistry. But the thing that really makes us unique, I think, is the fact that we have pharmacology in the drug discovery unit. And they are the final arbiters and really help accelerate the process. Because when I started in this, I would write to pharmaceutical companies, ask for compounds, then test them perhaps in vitro, and then walk up to an animal and had no idea how much to give. And if nothing happened, I didn't know why. Whereas with pharmacology, you do know, because we can accurately predict the dose, the duration, the route, etc., etc. So that's been extremely helpful. So we have three approaches to drug discovery that we use. I can't say one is better than the other, basically. I think I've got examples following showing a target-based, a phenotypic, and a drug repurposing strategy where you can either start with a fully validated target that is well characterized and genetically shown to be essential for survival of the pathogen, and then carry out screens to generate hits and then test them against in cellular models, and follow through hit validation and so on into leads, and then lead optimization. Likewise, you can start with a phenotypic hit, and you've immediately got round some of the problems we've run into, which is the fact that compounds don't necessarily get into the cell and give you the expected pharmacological result for whatever reason. And so that deals with some of the key druggability issues using the phenotypic approach. Now, not everything gets through to preclinical candidate, but as you can see, there are outputs on the bottom where chemically validated target and tool compounds can be obtained. And if that comes from a phenotypic screen, we can then go back to identify the targets. And that is an interesting academic challenge for the basic scientists in the team. Likewise, not all molecules will be suitable at the animal stage, and there you can say, well, we have a pharmacologically validated target. It may not be suitable for development into human disease, but nonetheless can be a driver for future development. So industry partnerships, our strongest partnership is with GlaxoSmithKline and Tres Cantos and the kinetoplastid DPU there. I've just come from our annual Scientific Advisory Committee meeting, and I'm pleased to say that we have a new drug potential for visceral leishmaniasis, which is now going into preclinical candidate selection. We have 4 candidates, and we're working out which one would be best. With HIT-TB, this is a newer collaboration, but we have screening compound sets from a wide variety of different pharmaceutical companies. AbbVie's interested in supporting safety pharmacology, and we're also working with GSK on certain common chemical series that have been identified. In addition, we're working with Eli Lilly on lead optimization of one of their compound series. And in terms of deconvoluting targets from phenotypic screens, we're actively involved with CellZone. So I'll give you 3 examples very quickly. First of all, one of the target-based approaches that we started with, we started with 25 targets scheduled for entry into drug discovery, and through a process of attrition, we ended up with one that got into a full lead optimization, and this was N-myristoyl transferase. But before we took it on, it had only been validated in vitro, so experiments were done in our university to show that it was essential for viability in vivo. High-throughput screen was developed, and a number of hit series were identified, but particularly a pyrazole sulfonamide series, which, after some extensive and skillful medicinal chemistry, led to the compound on the right of the screen there, DD85646, which had nanomolar potency against the target and against the cell. As you can see from the structure-activity relationships within this particular series, there was a good correlation between target potency and cell potency, and we were able to show that this was the mode of action in the trypanosome itself, and that it was a cidal compound. We were going great guns. This was only 2 years into our project, and we had curative activity in the acute model of sleeping sickness. That's before the CNS is invaded. And that's when we ran into a problem. That WHO, Department of Neglected Tropical Diseases, changed the target product profile of the drugs they were looking for to treat human African trypanosomiasis to anything by any route that would treat the second stage of the disease when the brains become infected. So I think we burnt another 18 months on this, trying to get the appropriate pharmacological parameters. If we made the molecule smaller and we got better blood-brain barrier penetration, we got weaker activity, and so it went on. And so this molecule is parked in the sense that we're very happy to partner with anyone who would like to either take on the target, okay, to find a new start point for the N-myristoyl transferase as a potential target for sleeping sickness. And I think the problem is you've got to get molecules that cross the blood-brain barrier. So let's move to another one. This is only just been approved for me to show, I think, on Monday. And this is a new anti-malarial with a completely novel mode of action. And this was a phenotypic screening approach, starting with our in-house protein kinase inhibitor set, which showed potency, validation, 10-point potency curves. And the 120 hits were then sorted into 14 series. Talking with Medicines for Malaria Venture, they said only 3 of our 14 series were not being worked on by others. And of those 3 that we took on, 2 were very quickly got rid of, and we were left with MMV04 series. So through lead optimization around the core structure, the compound shown on the right, DDD 107498, was arrived at, and that's extremely potent against Plasmodium falciparum blood stage, EC50 less than 1 nanomolar, including all known malaria-resistant lines, and also being tested against several hundred clinical isolates. The next stage was to find, to our delight, that this compound is actually active against multiple life cycle stages, both the liver, the blood, gametocytes, and even if you feed mosquitoes with infected blood. So this has the possibility to have prophylactic activity as well as transmission blocking activity associated with treating the disease stage, the erythrocytic stage. The compound has excellent oral PK with a long half-life varying as you go from mouse to rat to dog to monkey, from 10 to 29 hours. It's active in the GSK SCID mouse model of human malaria with an ED90 of 0.6 milligrams per kilogram for 4 days. And it's now under preclinical development in partnership with Medicines for Malaria Venture. And the predicted single-dose treatment, because MMV has a very high bar to achieve, will be 7 to 750 milligrams, and its predicted half-life is 45 hours. Now, we did the chemistry and we did the in vitro screening, but I'd like to acknowledge the fact that this was a huge multidisciplinary effort involving MMV coordinating activities with Monash University, Columbia, the universities of South Florida, California, Washington, as well as Dundee, Imperial College, Swiss Tropical and Public Health Institute, and Sanger Institute. So that's an example of disseminated collaboration in order to develop a new drug. This one really falls under the category of drug repurposing. So fexinidazole was of interest and is of interest in DNDi for the treatment of human African trypanosomiasis. But it had been tested against leishmaniasis and found to be inactive, and also inactive in animal models if given intraperitoneally. But we'd been working on drug resistance in sleeping sickness, and we knew that fexinidazole is metabolized to first the sulfoxide with 1 oxygen and then sulfone here. And the mode of action we understood was through biological reduction of this nitro drug to cause toxicity to the African trypanosome. So my chemist, I have one chemist in my own basic research team, synthesized the 2 metabolites, and we were delighted to find they were active against leishmaniasis. And working with the drug discovery unit, Kevin Reed's DMPK team, we were able to show in the lower quadrant there that actually it was the sulfoxide and then the sulfone that combined, shown here, give adequate coverage for 24 hours with a single oral dose. Fexinidazole, of course, is inactive, and as you can see, hardly appears on the screen at all. So then we did animal studies compared to pentastam and miltefosine, found a good dose-response relationship. And this is when we were able to hand this over to DNDi because they had already established that it had good oral pharmacokinetic behavior in phase 1 trials with African subjects, I think carried out in Paris. And the predicted pharmacological behavior and the data we had produced have encouraged them now to commence phase 2 trials in Sudan. And I believe they're still recruiting, but hopefully somebody can tell me if I'm wrong. But this is an example of drug repurposing, basically. It was intended for one use, and now we found another. So we have other things in the pipeline. I've mentioned the N-myristoyl transferase inhibitors. Some of our failed projects for human African trypanosomiasis have now been repurposed for the treatment of animal trypanosomiasis, or Nagana, and they're in veterinary trials in Burkina Faso, supported by another PDP, GalvMed. I've mentioned the candidate selection for the BA05 with GSK, and we have a lot of promising potential drugs for Chagas disease that do not operate via inhibition of sterol biosynthesis, because I'm sure many are aware that two CYP51 inhibitors have failed in clinical trials. So what do I think's needed for innovation? We need a driver, unmet medical need in orphan and neglected diseases. We need to combine basic science with drug discovery know-how, and we have to be prepared to take risks. Novel approaches require risk. We think we can do that as academics. And obviously, we work very much in a multidisciplinary environment. You need strong institutional support, and the University of Dundee has committed a huge amount to providing us with the buildings and infrastructure to be able to establish this. And of course, none of this can be done without extensive funding from the Wellcome Trust and other charities, PDPs, and industrial and government funding. So I'd just like to thank you all and just say that I believe science is the most communal of human endeavors, and to acknowledge the team who's really done this work. It's not me, it's the chemistry teams and the biology teams and the DMPK teams who've been critical to the success so far. And I'd like to acknowledge the very productive partnership that we have with GlaxoSmithKline at Tres Cantos. Thank you very much.